Hey everybody, Mr. J here. Today we are going to be going through cells, the base unit of your body. If you were to guess, take a count. How many cells do you think you have in your body? Okay, if you guess like, I don't know, 1,000, maybe 10,000, you have 30 trillion of your cells in your body, and more than that, you probably have about 100 trillion bacterial cells in your body as well. So you are made of cells. So we're going to discuss everything you need to know about cells. And I also want you to check out the other two videos about membrane-bound organelles, as well as cell transport. That'll be very helpful, kind of putting things into a concise manner. This is going to be more of a big overview. So definitely watch those two videos and then go through this entire thing. It'll help you tremendously for the rest of the course. Trust me on that. So here are your learning objectives. You can look over them real quick. We're basically going to talk about cell structure and function, what makes them different from each other, like how many different types of cells do we have and what do they have differently. We're going to talk about the outside of the cell, so the cell membrane, and why it's so important to everything you do. You're going to hear about cell membranes a ton in this class. We'll briefly go through organelles, which means baby organ, so little organs of the cells, including several of those like the nucleus, cilia, flagella, only a few have those. And then we're going to talk about two other things, which is the cell cycle in stem cells, and then cell transport. So again, we're going through all of these in order, so you can kind of check them off as we go. So let's get started. First off, no two cells in your body are exactly alike. They're insanely similar, but they have different structures and different functions. So if you look at these three, this guy is called a nerve cell or a neuron. These can be up to 3 feet long because it's traveling from like your brain potentially down to your hip, okay, to move like a hip muscle. So they can be super long neurons. So the structure of the neuron is a long winding road that looks like a cord, fits the function to send a signal from, say, brain down to a muscle cell, okay? Here, these are epithelial tissue, which means sheet-like, okay? So it's very thin cells. This allows things to pass in and out of the cells very easily, and it's also a nice protective covering. So your skin, for example, the outside of your skin is made of many sheet-like layers of these cells called stratified squamous epithelial tissue. Basically means a bunch of sheets on top of each other. And then lastly, this is your muscle cells, specifically your skeletal muscle. Why? Because they've got these little ridges, kind of line-like structures, and these all contract. So all of those units contract, and it shortens your muscle. So again, you see how the structure of the cell determines the function depending on what it looks like, right? So you've probably seen this composite cell, the general cell in your biology classes, right? This doesn't exist. Okay, this cell does not exist in your body, nor does it exist anywhere else, really. This is just showing you all the possible things that are in the cells themselves. So we're going to go through a few of them, not all of them, but I'm going to go through the most important ones. So all of these different organelles, as well as the cell membrane. So let's start at the cell membrane. I want you to think of the cell membrane as the control center of the cell. You probably thought that was the nucleus, right? But really, the cell membrane, the thing that allows things in, puts things out, kind of talks to other things, that is the control center. So look at how many things are in the cell membrane. There's a few things I want to point out. Think back to your macromolecules. We talked about probably 3 main ones, right? Proteins, carbohydrates, and lipids. Well, if you look at the cell membrane, the main part of the cell membrane you may have heard of as a phospholipid bilayer. So here's your phospholipid bilayer. What does it mean? Phosphate groups are the circles. These like water. And the lipid in the center. These don't like water. So why are they organized in a bilayer? Well, because the inside of your cell, that's this region, and the outside of your cell, which is this region called extracellular, these are fluid-based, water-based, okay? So the outside of both sides of the membrane, both in and outside of the cell, need to have something that can associate with the water. So that's why you have the polar ends. However, or the polar regions of the phosphates, right? These are the phosphates, the circles. The lipid bilayer, the lipid section in the center, this allows cholesterol to be packed in here. It allows some structure of the cell so it's not easily breakable. So these are things that are made of lipids, will be found in the center layer of the phospholipid bilayer. Now, key point, you're going to remember this later, the things that can pass easily in and out of the cell are going to be lipids. Interestingly enough, lipids can pass right through the bilayer, whereas things like Water, other polar molecules, maybe like sugars, carbohydrates, they can't pass through very easily. So we're going to talk about that later. So let's talk about the rest of these structures first, and then we'll get to that later. So these big hunks of stuff, these are proteins. These are things your cells made themselves and embedded them into the cell membrane. They can do several different things. If you look at this guy, it's got a little chain of carbohydrates. These would be little glucose molecules attached to the protein. These are cell identifiers. They are called glycoproteins. Glyco meaning sugar, protein meaning protein. And it basically tells things outside the cell like, hey, this is who I am. I am a part of the body. Don't kill me, please. Okay. So they're identifying markers. Okay. These are integral proteins, these little green ones. Okay. They just give structure to the cell. They don't do a whole lot. But these guys are channel proteins. You can guess what these do. They let certain things in or out. And each channel protein is very specific to what it's letting in or out. So for example, you've got channel proteins called aquaporins, which means water hole. They allow water to pass through in and out easily. Okay, because water, like I said earlier, is a polar molecule. It doesn't like getting through the lipids. Water doesn't mix with fat, right? So you need to have a channel for it to pass in and out. Okay. I also talked about carbohydrate chains already. These can be cell identifying markers as well. And then sometimes you have what's called receptor proteins, okay? Receptor proteins are going to receive some sort of signal that will bind either to like the sugar part or the protein part, depending on what it is, and it'll tell the inside of the cell to do something. So it's almost like you're receiving a message here and you're doing something on the inside here. Okay, think about it like your Ring doorbell, okay? Like somebody comes to give you some mail on the outside of the cell, and then they ring the doorbell. That triggers you on the inside of the house to go do something, to go answer the door perhaps, okay? So it's a signal coming from the outside, and then the inside of the cell does something different, okay? And then also we've got some cholesterol in here. I mentioned that earlier. Cholesterol is a fat, so it fits nicely inside the lipid bilayer. So as you can see, the plasma membrane is not just made of those phospholipids that you learned about in biology class. They have a ton of stuff packed into it that do a whole lot of different functions, and that is why I call it the control center of the cell. So I just talked you through all of these, so we don't even need to talk about these. Now let's talk about the organelles, okay? So organelles, if we go back to this general cell, all of these organelles, I want you to remember, have a phospholipid bilayer. They have a membrane around it. Why do they need a membrane around each of these individual organs of the cell? Because all of them are doing different things. They're all compartmentalized. Okay, think about them as different rooms of the house. Each room has a specific function. Okay, so you don't cook in your living room. Okay, and you don't watch TV in your kitchen, and you don't sleep, well, usually in your dining room, right? All of them have a certain function, so they have to be sectioned off from each other. Okay, think of all the cell's organelles, they're tiny little organs inside of here, as compartmentalized because they have that membrane around them so that they can do their own thing, their own chemical reactions within them without bothering any of the other organelles. Okay, so I'm going to talk through a few of them that you'll see several times. Endoplasmic reticulum. Big long word, just call it the ER, okay, the ER, right? The ER is very long and spread out, and it's got, if it's a rough ER, it's got a lot of dots here, okay? And then you also have some that are smooth, so the smooth ER that doesn't have the dots. Well, what are the dots? That's going to be key to what the ER does. These are ribosomes, okay? You've heard of ribosomes before. I think of ribs in here because ribs are protein-based, right? So you eat ribs, they're protein. Ribosomes make protein. They're little chefs, okay, that take information from the DNA of the cell, the mRNA, and they read it and they make little proteins out of them, okay? Those proteins build your cells, they build your whole body, okay? So the ER does a really good job of packaging these proteins for wherever they need to go in the cell. They might need to go to the cell membrane. They might need to stay within that area. They might need to go to the nucleus. So the ER is just packaging them up, putting them where they need to go. The smooth endoplasmic reticulum doesn't have ribosomes, so they don't make proteins, but they modify them. They add things to it. Maybe they add like a glucose chain, like we saw here. These little protein carbohydrate chains originated in the endoplasmic reticulum of the cell. So eventually got transported into the cell membrane and got embedded there. So this is kind of like the packaging office, the post office of the cell you may have heard of before. And I want you to give you an example of your body. So not every cell in your body has every single organelle. And I'll say that again because that's a key point. Not every cell in your body has every single organelle. The endoplasmic reticulum is especially common in your liver. Why? Well, whoops. In your liver. Why is it so common in your liver? Your liver makes so many different things called enzymes, proteins, that help do different chemical reactions. They detoxify things that you put into your body that were bad. They modify things that are good so that they can be even better. They do all these things with these proteins that they're making. Where are the proteins made mostly? At the ribosomes in the endoplasmic reticulum. So if you look at a liver cell, if you look at a liver cell and you look inside, you will see all sorts of these endoplasmic reticulum branches, okay? Structure fits function, so if it needs a lot of protein, it's going to have a lot of endoplasmic reticulum. All right, let's move on. That's plenty of endoplasmic reticulum. Golgi apparatus, I'm just going to say it's basically the post office part of the endoplasmic reticulum. So the proteins are made, they get sent here, they get packaged up for transport, maybe out of the cell, maybe to stay in the cell. They're just packaging things and putting them places. Okay, so about the same function. Mitochondria is the blank of the cell. You probably said powerhouse, right? You learned that in biology day 1. And that's the only thing you remember. But the mitochondria is technically the powerhouse of the cell. Why? Because it makes ATP. And ATP is the energy currency molecule for your body, for your cells. You have to convert everything from sugar to fat to ATP so that you can use that to then do what you need to do in the cell. Think about it like if you are from Europe and you're using the euro currency, okay, that's going to be your sugar, okay? Now you go to the United States. Well, you have to take all that those euros and convert them to dollars, US dollar, ATP. So now you can use it to actually buy things, right? So it's just a conversion into ATP because that's what we use in the cells. And that's what the mitochondria does. It does a lot of other things I'm not going to get into, but mitochondria is a fascinating thing. Okay, a few other things. Whenever you see lysosomes or peroxisomes, lyse and peroxide kind of sound like chemicals that break things down. So to lyse something means to burst. These guys like to recycle things within the cell, proteins, carbohydrates, all the same. And then you also have the cytoskeleton, which is like the structural units of the cell. So it's like the scaffolding. It provides structure support. And then you got a couple other things. So let's point these out. Cytoskeleton kind of sounds like cell skeleton, which it does. It's just the skeleton of the cell. It provides support to the cell. Centrosome, you probably won't run into all that much. These guys are eventually going to form the spindle apparatus that divides the cell into two during mitosis. We'll talk about those later. Cilia and flagella. This is a really good example when I said that not all cells have the same organelles. So let's consider these organelles just for simple fact, okay? Cilia, these look like little, if you've ever seen kelp like blowing in the ocean, blowing, that's probably the wrong word, but like flowing in the ocean, doing this sort of thing, right? These are the same things inside your body. They're these little hair-like projections that beat basically the fluid or air some direction. These are common in your respiratory tract, okay? They're especially common when you breathe in and there's some gunk in the air and then you start coughing. Why? The cilia caught all that dust and bacteria and crud, and it moved it up from your lungs up to about here. So then it triggers a cough reflex to get that crap out, because otherwise it could harm you. Okay, so that's a function of the cilia. This is flagella. Really the only cell in the human body, and males specifically, are the sperm cell, and the flagella are basically the whip-like tails that help propel the sperm as it's swimming. Awesome. Cell nucleus, another membrane-bound organelle, actually a double-layered membrane-bound organelle, so it's very, very protected. This is like a Area 51, Fort Knox vibe, okay? Not many things can get in, not many things can get out. This houses your DNA, okay? Your DNA, think of it as the massive instruction manual to build your cells, because DNA will eventually code for the proteins, which I showed you. There are tons, all of these things that I said, like the cytoskeleton, all the membrane proteins, all of these had to be produced by your cells. Well, the instructions on how to produce them were found in the nucleus, okay? So how did it get there? Well, nucleus is made of DNA. DNA is transcribed onto mRNA, which is like the transcript or the recipe. It gets sent out of the nucleus, goes to the ribosome to be produced as a protein, okay? So the ribosomes use the mRNA as a recipe to build the protein. We'll talk more about that in next chapter, chapter 4. But this is why it's called the control center, because it's kind of saying like, hey, make this protein. This is how you make this protein. This is how you make this protein. Think of it the blueprints of the cell. All right, you can copy these tables. It's pretty good structure function of each cell cell organelle, but I'm just going to skip them for now. There's several other ones I did not talk about, but they're not as important to this class, so I'm going to skip that. All right, so pause for a second. Think about what you just learned, maybe write a few things down, and then we're going to move into cell transport. Okay, so cell transport. This is talking about things that are getting into and out of the cell. Okay, how do we do that? Well, we've got a nice bodyguard in the cell membrane, right? So it's filled with a phospholipid bilayer that allows lipids in easily, but not other things like sugars and water and stuff like that. And we also have these little channels that can open and close or stay open that allows things in and out. So this is why I call the cell membrane the control center, because it's allowing basically whatever you need to in and whatever you need to transport out. Okay, now the motion itself is either passive or active. If you hear it's passive transport, that means it doesn't require energy. It passively moves from high to low concentration. So high to low concentration, if there's a lot of stuff here, a little stuff here, it's going to move towards where the little stuff is. Okay, let me give you an example. If you have Axe body spray, so go back to your middle school days, there's a high concentration of Axe in the body, in the can itself, right? So when you press the lever, it opens up the hole, and then all of those aerosol molecules are going to go out. Well, if I spray it here and my colleagues across the way, there's a high concentration right now of molecules that smell like Axe right here, and eventually they will move everywhere else. There's low concentrations of Axe. So high concentration to low concentration everywhere else. And eventually it'll go all the way across the hallway and get to my colleague, and they will yell at me because they're like, why did you just spray Axe body spray in your office? Well, I was showing a point. So it's just high to low concentration. I didn't use any energy for that. It just naturally wanted to do that. That's just the nature of things, to move from high to low concentration. If you don't believe me, things in nature move from high to low. Gravity, right? High to low. So that's passive transport. Active transport is when we use ATP energy to move things from low to high concentration. Why? Because that's not natural. Moving things from low to high concentration is not natural. We need to use energy for that. Okay, think about like if you're carrying something up a mountain, right? You have to use your energy to walk up the mountain. Whereas passive transport would be you let that thing go and it just rolls down the mountain naturally, right? So that's passive versus active transport. So here's another example of sugar dissolving. There's a lot of concentration of sugar here, and it'll all dissolve equally out through the water. That's just simple diffusion, high to low concentration. Here's an example of it going across a membrane. So permeable means permissible. So is it allowing something in or out? So in this case, I have a membrane that allows both water and a solute, which is just something dissolved. We'll say it's salt or something like that, okay? So salt, sodium chloride. So we've got a lot of salt here. We've got a lot of water here. Well, the water will want to move where there is less water. The salt will want to move to where there is less salt. So we look at this, and the water is going to want to move left, the salt's going to want to move right, and eventually it'll be equaled out on both sides. Okay, so it follows its own concentration gradient. What do I mean by that? Water only deals with water. If there's more water here, less water here, it's going to want to move to where there's less water. Okay, if salt is high here, no salt here, salt's only going to deal with where there's less salt. Okay, so high salt concentration is going to move to low salt concentration. They don't follow each other's gradients. They just focus on their own. So water follows low water, high salt follows low salt. We see this in the body. We see this in the body a lot. So in your bloodstream, when you breathe in, you're basically putting a bunch of oxygen into your blood vessels. Okay, There's high concentration of oxygen in your blood vessels then, and you feed that oxygen to your cells. So oxygen just diffuses from high concentration to low concentration because there's less oxygen in your cells. And in your cells, there's a lot of carbon dioxide. But in your bloodstream, there's low carbon dioxide. So then the carbon dioxide gets pushed out of your cells, high to low concentration. And and that's all I wanted to say. So carbon dioxide will flow down its concentration gradient from high CO2 to low CO2. Okay, so that's a great example of diffusion in your body. Awesome. There's also things called facilitated diffusion. This just means those things that are dissolved need a protein. A protein in this example, the carrier protein, channel protein, sorry, these are channel proteins. They're just like, think about them as doorways, okay? I open the door and I let something bigger in or out that I couldn't get through the small like mail hole, perhaps, okay? So these are just when things are too big. So like there's a higher concentration of something out here, low concentration here. But since it's so big, it can't pass through easily. So it needs a big garage door to allow it in. And that's through these channel or transport proteins. So here's an example of being selectively permeable via osmosis. Selectively permeable, what do I mean? Only the water molecules here are going to pass through. The proteins, these big ones, are not going to move. So check out what happens. I put the membrane in, there's equal amounts of water, there's more protein here, okay, which means there's less water dissolved because the protein's like kind of keeping the water levels low. And there's low protein here, high water. So water is going to follow its concentration gradient. So there's high concentration of water here, lower concentration of water here. So where's water going to move? Into A. What's going to happen? Water will actually build up on this side and the protein will stay on the right side. So if you count the proteins on the right side, 1, 2, 3, 4, it's the same as here, 1, 2, 3, 4, and the same on the left side if you were to count. The water molecules were the only things that were moving. That is called osmosis, the movement of water across a semi-permeable membrane. Okay, hopefully that's making sense because now I'm going to get into an example. You've probably heard of these solutions before, hypo, hyper, and isotonic solutions. This just means that water is going to either move out, stay the same, or move into the cell depending on where there's less water. So hypotonic solution. This means there's a lot of water here on the outside of the cell, and there's less water inside of the cell. So what does that mean? The blood cell has more stuff dissolved in it, and the stuff outside does not. So there's more water outside, less water inside. Where's water going to move? Into the cell, and the cell will actually bulge up. So this is a red blood cell. It's supposed to look like this, but it bulges up because water moved into it. You may hear this in another section or another video. Water likes to follow salt. Now, that doesn't mean it's following the salt's concentration gradient, but what it means is that where there's salt, there's less water. Where there is salt, there's less water, okay? So where there's salt inside the cell, there's less water. So there's a concentration gradient, high water to low water, hypotonic. Isotonic, this looks normal. This looks normal. That means that there's equal amounts of movement into and out of the cell. So that means there's equal amounts of water concentration on the outside and inside the cell, so it doesn't change at all. In your body, you are at about 0.9 sodium chloride solution. Don't worry if you don't know what that is. If you're a nurse, you put in a saline solution, it's 0.9% sodium chloride. That is isotonic, equal in concentration to your cells, your red blood cells, as is the solution you're putting into the veins, okay? So that means it's isotonic, so no total water movement will go in or out, okay? Equal balance. Lastly, hypertonic solution. That means there's more salt outside of the cell, so that means there's less water outside of the cell. Therefore, water will move from where there's high water inside the cell to low water outside the cell. Or you could say it this way. The cell is less salty than outside. Therefore, it's going to follow where there is more salt, okay? Less water. I hope I'm not confusing you by saying that. Water follows salt because where there's salt, there's less water. So therefore, water will follow its concentration from high to low, wherever that may be. Okay, So now this is some examples of just filtration through the capillary walls. So I'm talking about individual cell membranes like this little section here. So this is the outside of this cell here. But sometimes in your capillaries, this is where your blood vessels get really, really, really tiny and they start giving other cells oxygen, carbon dioxide, nutrients, that type of thing. There are tiny little holes. They're called fenestra, which means window. And that just allows things to pass into and out of the cell easier. So it kind of squeaks fluid through here and there. That's not as important. So now let's talk about active transport. All the stuff we have been talking about is high to low, passive, no energy, right? Active transport, I told you, is pumping things from low to high concentration. Well, what is that example of? Well, there's a sodium-potassium pump. That's going to be important in your neurons and a whole lot of other things. But it happens when there's a lot of sodium outside the cell and a lot of potassium inside of the cell. So we're going to use a pump to amplify the difference. What do I mean by that? Okay, we're going to pump sodium from low sodium to high sodium. We're going to pump potassium from low potassium to high potassium. Why is that important? The cell inside likes to have a lot of potassium. It wants to keep the sodium outside of it, okay? Just for some reason or another. We'll go back to this later on in this class. But it likes to keep sodium out, likes to keep potassium in. How does it amplify that? Sodium-potassium pump, pumping sodium low to high concentration and potassium low to high concentrations with the use of ATP, that energy molecule, right? So we'll get back to that. We will get back to that later. I can't talk. The other two forms of active transport are endo and exocytosis. You may have heard of phagocytosis, basically the same thing as endocytosis. It's when the cell, like a white blood cell, eats something, maybe a bacteria, right? It brings it inside of the cell membrane by engulfing it, okay? This is an example of a cell eating basically a lipid, or a liquid droplet, it looks like, in a different particle, maybe that's the bacteria, and it just engulfs it, okay? So it basically moves its cell membrane around it, engulfs it, and then makes that its new cell membrane. So just engulfs it, kind of brings it into itself. That does take energy. And then the opposite is also true. You can also have exo, which means out of the cell, exocytosis, and it can push it out of the cell. So large transport out of the cell. I won't talk about receptor-mediated endocytosis. It just is like a different mechanism by which you bring things in. We won't talk about that all that much. So here are some summaries of the different active and passive transports. You can screenshot these if they're helpful for you. I think the diagrams are a little better, so screenshot those. But here you go. Here are the charts. I won't go through them. Last thing, okay, last thing is a cell cycle. Cell cycle, this is just the lifespan of your typical cell, okay? So let me give you a fun fact. Every 7 years, you are a completely new person. What do I mean by that? Approximately all of your cells in your body have replicated, okay? So you've made basically from 1 cell to 2 cells, and then this old cell maybe died off, and now you have the new cell. So every 7 years, all of the cells that you had that first start of the 7 years are different than the cells you have after 7 years. Most of them. That's not completely true because some neurons stay there. But anyway, you have to basically regenerate your cells every so often to keep them fresh, to keep them working properly. This is just the standard lifespan of the cell. So the first main part is called interphase. That's the biggest part. And there's a period of growth called G1. G stands for growth. S, where you're replicating your DNA, preparing it for division. So when you replicate it, you have enough for both cells. Then you've got another growth phase where you're building up more organelles, preparing it for division. And then you have what's called mitosis, which literally means the splitting of the DNA or the cell itself. This has 4 stages called prophase, metaphase, anaphase, and telophase, followed by cytokinesis, which means cell movement, to basically go from cell 1 to 2 cells splitting apart. Okay, so those are the main phases of the cell cycle. Mitosis, they go through pretty quickly. I said prophase, metaphase, anaphase, telophase, which is PMAT. They look differently as to what's happening in the cell. So if you look at it, let's start here in prophase. You see the DNA is condensing into chromosomes. We'll talk about this more in chapter 4, I think it is. They're condensing. In metaphase, that's next, they a line in the middle, okay? In anaphase, they begin splitting apart. So basically what's happening is you duplicate your DNA, right? Preparing it for the cell to divide and have two separate cells with fully functional DNA. So you package them into the chromosomes, you put them into the middle, and now you're going to separate one half from the other half so they both have complete sets of DNA. So anaphase is when they're starting to split, and then telophase is when they're starting to get to the other ends, and the DNA in the chromosomes are starting to unravel. Okay, so now you see instead of being packaged all nicely like this, now they're starting to unravel and then eventually divide, cytokinesis. So now you have two fully functioning cells. Now, what happens a lot is in, for example, the base of your skin, you keep regenerating skin, you slough off your skin like thousands and thousands every like minute, which is kind of gross if you think about it. But as they divide, so say this is the base layer of your skin right here. Here's a cell. This is going to be what's called a stem cell. It can regenerate constantly and constantly and constantly. This stem cell can replicate, okay, and now I've got two cells. This is going to remain a stem cell, but this cell is going to get pushed up to the top of the skin layer. Okay, so these guys will eventually die off, but you still have the stem cell that can keep dividing and keep dividing and keep dividing to make more and more and more and more skin cells as they die off and slough off. Okay, so that is shown here where you have a stem cell dividing to something that will do something, but then it also remains as a stem cell. So one stays as a stem cell, one goes on to do something. It could be a gland cell, it could be an epithelial cell, that would be your skin. It can go and turn into a neuron, it can go and turn into a different cell. So these stem cells, a lot of them can turn into different cell types, but majorly most stem cells just turn into one main cell type at this point in your life. Now, when you were an embryo, this is way more than you need to know. When you were an embryo, like a single-celled organism at one point, right? Then that divided into two, those are considered stem cells because eventually they'll turn into a neuron, like a brain cell. They'll turn into a skin cell, a muscle cell, epithelial cell, a cell that has cilia on it, all these different cell types, your liver cell, heart cell. Those would be called pluripotent. They have a lot of different possibilities to turn into, but most stem cells, when we're talking about your body right now, they're just going to turn into one cell type or another. Okay, hopefully that made sense. They're just undifferentiated. They can turn into a specific type. Last thing, cell death is called apoptosis. Some people may say apoptosis. I've heard it said millions of different ways. This is just programmed cell death. Why would this happen? Well, sometimes your cells get sick in a way. So maybe they screw up their DNA. They're making more DNA and it gets screwed up, and now the cell is going to really wreak havoc on your body, maybe turn into cancer. A lot of the times your cells will have checks on themselves to be like, we didn't do that right. We should probably just kill this cell off so it doesn't ruin the rest of the body. So that's called programmed cell death, otherwise known as apoptosis. Okay, so we'll talk about that a little more when we talk about cancer and other diseases at the end of the semester, but that is it. If you have watched this entire thing, I am so proud of you, first off, and I hope this was helpful. Hopefully it was a good overview, and please let me know if you have questions in the comments below. Be sure to like and subscribe if this helped you.